If you are anything like me, you'd hate going to the hospital to have your bloods drawn. Now imagine this. If all your medical checkup could be completed just by doing this. Done. That's the promise of exhale breath condensate, or EBC in short. We collect the breath, condense them into small droplets, and observe the chemistry inside them. They contain VOC, or volatile organic compounds, which carries important information about what's happening in our body. Now, why haven't we seen them more in clinics, you may ask? That's because the results obtained from EBC varies massively. The same biomarker in one study may show very high concentration, whereas in another study, it may show very low concentration. This variation makes it very difficult to use EBC as a reliable source of clinical biomarker. We suspected something. Maybe it's not just the physics, it's drawing up this conclusion. Maybe it's the droplet physics. In order to answer our question, we developed an experimental setup using fluorescence microscopy. Essentially what we did was we lit up those VOCs in the droplet so that we can track them in real time during evaporation and condensation. And what we saw completely changed how we view EBC in clinical scenario. Not all droplets are equal. That means some droplet may contain large amount of VOCs, whereas another droplet may contain none of it at all. So, if our sample is filled with the wrong type of droplet, we may have lots of water in our sample, but not a single trace of biomarker inside. The second question we asked ourselves was, why do some droplets evaporate slower compared to other droplets? The key finding here was the suppression of evaporation. At low concentration, the VOCs inside the droplet struggle to come out of the droplet. Interestingly, this phenomena was not caused by the liquid in the droplet, but rather the air surrounding the droplet. This air creates a small, thin layer of invisible barrier that essentially traps and prevents the VOC from coming out, causing slowed evaporation in the process. Now, what does all of this mean to us clinically? It means that if we understand the physics behind a droplet, we can control it, model it, and predict it. We can develop an experimental setup with controlled temperature, humidity, and airflow that can maximize the collection of our samples from exhale breath. As such, with the research going on, hopefully the day is not that long when we can walk out of a clinic with one less bandage in our hand. Thank you.